Okay, in today's video notes, we're going to talk about the structure of DNA, and then we're also going to start talking about DNA replication. We're going to try to cover all of that in this single video. So I'm going to go a little bit quicker than normal in hopes of getting everything we need to. So with DNA, okay, DNA is made up of three different things, and we can basically call each of those units a nucleotide. And a nucleotide contains three parts. So the first of which is the 5-carbon sugar deoxyribose. Second of which is a phosphate group. Those two materials make up the sugar phosphate backbone, or the outside portion of the overall DNA strand. And then the inner portion, like the rungs of a ladder, would be the nitrogen bases, which are adenine, thymine, cytosine, and guanine. And so here we can see those three parts of the nucleotide. Once again, the phosphate group, the pentose sugar, and then the nitrogen base. And with the strand of DNA, that sugar, pentose just meaning it's 5 carbons or 5-carbon structure. The sugar in DNA is deoxyribose. If you look at RNA, the sugar would be ribose. So there's 4 types of nitrogen bases. Adenine and guanine are purines, which just means that they have 2 rings, and a 2-ring structure will bond with a single-ring structure, which we call pyrimidine. So that would be cytosine and thymine. So again, adenine will always bond with thymine, which is this 2-ring structure bonding with this single-ring structure, and guanine will always bond with cytosine. So basically, with the way that these molecules attach together is using Chargaff's rule. Again, adenine with thymine, cytosine with guanine. That's all we're going to cover with that one. I think it's just the overall structure of DNA. So this is just a double helix model, flattened out, turned to its side, and we still get our sugar phosphate backbone on each side. We have our nitrogen bases along the middle, and then we have these hydrogen bonds that are attaching those together. So with the nitrogen bases, cytosine and guanine, they need 3 hydrogen bonds to attach them together. Adenine and thymine, they require 2 hydrogen bonds to attach those together. Chargaff's rule just tells us which ones are bonding with each. Thymine with adenine, cytosine with guanine. Quick review, you can look through these if you would like. I'm going to move forward into the main part of section 2, which is the process of DNA replication. So we know DNA is found inside the nucleus of eukaryotic cells. In a prokaryotic cell, they don't have a nucleus, so it's going to be found throughout the cytoplasm. Variety of different number of chromosomes depending on what type of organism it is that we're looking at. Eukaryotic cells being much more complex than a prokaryotic cell. And here we can kind of see the difference between those two. So E. coli, which is a prokaryotic cell, is going to be 4.5 million base pairs. So in humans, we're going to have a little over 4 billion base pairs. And then all of that has to fit into each individual cell. So the way that that works is DNA folds itself around histone proteins, coils itself up to fit inside the nucleus. And so here we can see our double helix form wrapping around the histone proteins. Those proteins then coil themselves up and continue to coil up until we create the chromosome or chromatin form of DNA, depending on what stage of the cell cycle the cell is currently in. Complementary strands just mean that these DNA strands on one side are the opposite of the other. This is the same idea as Chargaff's rule, where adenine is going to bond with thymine, cytosine with guanine. Now, if we only see half of the genetic information of a particular DNA strand, we can determine what the other side should look like. So with this strand, we can determine the complementary strand, and we would just know that adenine will always bond with thymine, and cytosine will bond with guanine to create the complementary or opposite base pairing. Now we're going to move into DNA replication, which is just making the copy of a DNA strand. And so remember, this process is going to be taking place during S phase of the cell cycle. So that's when DNA is going to be copied, getting it ready for cell division in the near future. The first thing that has to take place is to unwind that DNA so that the enzymes can have access to the nucleotides, or specifically the nitrogen base portion of the nucleotides, eventually start making that copy, because that's where the main component of the genetic information is held. And so the first thing that's going to help in unwrapping that DNA strand is an enzyme called helicase. And so this is responsible for unwinding and then breaking the hydrogen bonds that connect together the adenine and thymine, or cytosine and guanine. And when this helicase breaks those hydrogen bonds, and allows the DNA molecule or strand to expand and move away from each other, allowing for more room for our next enzyme to come in, which we call DNA polymerase. DNA polymerase is going to be attaching the new complementary strand on each side to create the two new strands of genetic information, the two new strands of DNA. And so if we continue on in that process, each side of that DNA strand has to occur in a specific direction, just because of how all of the molecules are arranged on the sugar phosphate backbone. And so those two strands, the one that is able to move in one continuous strand, we call the leading strand. That one will just follow the helicase all the way along, attaching those complementary nitrogen bases. And then the lagging strand is the one that will have to be done in smaller pieces, because it can only go in one direction and it can't follow that helicase. And so those small chunks on the lagging strand we call Okazaki fragments, after the Japanese scientists that first discovered those. And so just realize that they're kind of like, each side of that DNA strand is kind of like a one-way street. The molecule, the enzymes just can't attach and move in the opposite direction. And so if we look at this, here's our helicase, which is breaking the hydrogen bonds between the nitrogen bases as it's moving through that DNA strand. And on the leading strand here, we have a DNA polymerase that is able to just go in one continuous motion all the way down that DNA strand and create that complementary strand, or the new strand, in order to fully finish that DNA copy. The other one has to move in the opposite direction. So this polymerase is going to go basically from right to left, and it will attach a small chunk, and then a new DNA polymerase, as you see back here, will attach on again and continue to complete that next chunk. As this helicase opens up, it always has to go from right to left, so it'll just do smaller chunks to complete that portion of the DNA strand. And again, we call those Okazaki fragments because of the gentleman that discovered those. So when we complete our overall DNA, or our new strands of DNA, the new cell will end up having one old portion of DNA, which served as the template, and one new version that was attached on from the DNA polymerase. So they get a little bit, or a combination of those two things, to create the overall genetic information of that new cell when those cells split. All right. The last piece for section 2 is that this process of DNA replication normally goes off without making any mistakes, but occasionally mutations will occur. And so when this does occur, it will possibly mess up the type of information that is being held in that DNA. And so normally adenine will bond with thymine, cytosine will bond with guanine, but occasionally mistakes do happen. And mutations can cause serious effects for an individual. Sometimes it'll have no effect on the cell. It just depends on where the mutation occurs and what specific portion of information is affected by that. So what protein gets affected by it, any other issues that may arise from that mutation. We'll talk more about mutations as we move on to section 4. So that is it with DNA replication. And next video notes will cover RNA and creation of proteins.